Hi, my name is Angelo Chan, and in this video series, I will explain how next-gen sequencing data is commonly processed. The slides in this video can be downloaded in the links in the description below. Let us begin. I'll begin by giving some background information on the topic before explaining what the core pipeline is. I'll go over the pipeline in an increasing level of detail, how to get to the pipeline from alternate starting points, how to set up the pipeline, and then some additional technical background information. In the context of biology, sequencing is the process of figuring out the DNA sequence of a strand of DNA. Sequencing has been a thing since the early 1970s, but in the early 2000s, a series of new sequencing technologies became commercially available, which had exponentially more throughput than previous technologies. These are known as next-gen sequencing, or NGS. Now, there are specific scenarios in which some of the older sequencing technologies perform better than NGS, but at least for now, we're not going to cover that here. The main point I want to make is that, generally speaking, all NGS data undergoes the same initial processing procedure. This is what I refer to when I talk about the core pipeline, and this is what I'll be going over in this video series. There are cases where this core pipeline doesn't quite work, such as when the DNA has been modified in some way, but even then, the procedure for processing that kind of data is usually just a modified variant of the core pipeline. Hopefully, by understanding what the data looks like at each stage of the core pipeline, you'll be able to understand why it doesn't work for certain kinds of data, and how the pipeline would need to be tweaked for it to work. I will start by giving an overview of the pipeline before breaking it down in more detail. And then, as indicated in the table of contents, some parts of the pipeline will be expanded upon in a second level of detail. I organized the information this way for two reasons. Firstly, because I didn't want to bog you down with minutiae until I'd given you the big picture. That way, you can better understand how these small details fit into the bigger picture. And secondly, because the second level of detail is only essential for someone who wants to perform the analysis themselves. If you are working with a bioinformatician who does the actual analysis for you, and you just want a better idea of what they do, then the first level of detail will be enough to get you going. That being the case, you would still benefit from learning how it all works. Apart from the core pipeline, I'll also go over the setup process separately, since the setup only needs to be carried out once per genome assembly, whereas the core pipeline needs to be carried out from start to finish for every dataset analyzed. Now, for anyone hoping to do some next-gen sequencing experiments, this video series is primarily bioinformatics. I will be covering the biochemistry behind some of the wet lab processes, but only on a need-to-know basis in order to explain the rationale behind certain parts of the pipeline. If you want a detailed guide to the relevant wet lab protocols, such as how to prepare a biological sample for sequencing, this video series is not what you need. As the name suggests, I won't be covering the many different downstream analyses which can be performed once the data has gone through the core pipeline, since the workflow can diverge significantly. I am also going to assume that the organism from which the NGS data was obtained is one with an official downloadable genome assembly. If no genome assembly is available, you'll need to assemble one using de novo assembly before the core pipeline can be carried out, and I won't be discussing the de novo assembly process here. The last thing to mention before we begin is that at detail level 2, I'll discuss caveats and examples which are specific to the software programs I use or am familiar with. However, those steps in the pipeline don't necessarily have to be carried out using the programs I mentioned. Alternate options exist for every step of the core pipeline, so don't be surprised if you read papers which mention an entirely different suite of programs which haven't been mentioned in this video series. I also won't include any example code in this video series, since the goal is to explain how the core pipeline works on a conceptual level, and not to give explicit step-by-step instructions on how to execute it. Such an example, along with the beginner's guide to command line, can be found in separate files. Let's start with a brief overview of the whole pipeline. We start off with the original FASTQ files, which contain completely unprocessed sequencing data. The FASTQ files undergo pre-processing, producing data which is still in FASTQ format, but is more reliable after having undergone trimming and filtering. These sequencing reads are then aligned against the reference genome, producing a SAM file, which contains raw, unprocessed alignments. These alignments then undergo standard post-alignment processing to produce sorted, filtered, aligned reads, which are now ready for analyses which are more experiment-specific. Now, I know that, technically speaking, we might start with something that's even less processed than a raw FASTQ file. In that scenario, you'll have to derive a FASTQ file from that raw data. And that will be covered after we finish going over the core pipeline, so don't worry. Anyway, now that we've done the overview, let's go into a bit more detail. I'll start by explaining what FASTQ files are. FASTQ files store DNA sequence data along with the quality scores for each nucleotide. The format could theoretically work for amino acid sequences, but personally, I've never seen it used for that before. So in the context of this video series, I'll only talk about FASTQ as a file format for DNA sequences. In FASTQ files, each data entry represents a single sequence of DNA, known as a read, and consists of exactly 4 lines. The first line contains the read ID. The second line contains the ACGT nucleotide sequence. The third line is irrelevant and often left empty, and the fourth line contains the quality scores of the nucleotide sequence. The FASTQ file format doesn't have a way to store additional information like secondary structure or molecular modifications, but that kind of information doesn't exist in the NGS data anyway. Single-end sequencing will generate one FASTQ file per sample per flow cell, while paired-end sequencing produces a pair of FASTQ files per sample per flow cell, although sometimes that data is stored in a single merged FASTQ file. Paired-end sequencing produces results with higher mappability than single-end sequencing, but costs more. Some techniques or analysis methods explicitly need paired-end sequencing in order for it to work. The FASTQ files that can be used for this pipeline can either come directly from a sequencing experiment, from an online data repository, or both. It's possible to pull together the reads from paired-end and single-end sequencing. However, this can bias the data, so discretionary judgment is advised. Also worth noting is that a lot of bioinformatics software requires paired-end read data to be stored in 2 separate FASTQ files, one for each orientation. Therefore, it is recommended that paired-end data stored in a single file be separated into 2 files, one for each orientation. Okay, so now that we know what FASTQ files are, let's talk about the pre-processing which raw reads undergo before they're ready for alignment. There's adapter and primer trimming, quality control trimming, and the combining or splitting up of files for logistical reasons. These steps can be done in any order because the inputs and outputs are all FASTQ files. As long as the necessary trimming and filtering takes place, the resulting data will be ready for alignment. It should also be noted that some of these steps are not always necessary. Newer sequencing machines usually trim off sequencing artifacts and segregate any multiplexed biological samples into separate FASTQ files, saving us the trouble of having to do it ourselves. Combining multiple FASTQ files into one is only necessary when multiple flow cells were used for the same sample, and splitting up the reads into multiple files is only necessary if the alignment process is being parallelized by multithreading. The alignment process itself is fairly simple. The reads are mapped against the reference genome to see where in the genome these reads originated from. Technically speaking, the reads are aligned against the index of the reference genome. But fundamentally, that doesn't change the fact that we're mapping to see where in the reference genome these reads originated from. The mapping process produces a SAM file. So what is a SAM file? SAM stands for Sequence Alignment Map, and this is the file format used to store alignment results. Some alignment programs produce BAM files instead, but BAM files are just compressed SAM files. They take up less hard disk space on your computer, but they contain identical data organized in the same way as their uncompressed counterparts. I will explain SAM files in detail, but you can also refer to the official SAM file specifications document linked in the description below. The SAM file format can be described as a table with header comments. That is to say, a SAM file consists of two parts, a comments section and a data section. The comments section consists of lines which begin with an @ symbol and are typically used to record metadata such as the details of the alignment procedure, which was used to derive the alignment data. The data table contains the alignment data itself in a TSV format. TSV stands for tab separated values. So in raw data form, different rows of the table are separated by new line characters, while the cells in each row are separated by tab characters. Each row of the TSV section corresponds to the alignment results for a single read. The first column contains the read ID. The second column contains information on how the alignment went, and this information is stored as a number which uses the bitflag system. The third and fourth columns contain the chromosomal coordinates of the best mapping result. The fifth column contains the mapping quality score. The sixth column contains more information on how the alignment went in a cigar format. The seventh and eighth columns contain the chromosomal coordinates of the next read in the data. The 10th and 11th columns contain the DNA sequence and quality scores of the read respectively. These are not always identical to that of the corresponding entry in the original FASTQ file. Some SAM files have additional columns with additional information depending on which program generated them. The most important information lies in the third and fourth columns, which contains the chromosomal coordinates of the alignment. Okay, now that we know what SAM files are, we can talk about the standard post-alignment processing steps in the pipeline. These steps are fairly standard across almost all NGS data, because some sequencing and biological artifacts are almost ubiquitous across different NGS techniques and technologies. The steps are as follows. Firstly, if the reads were split up for parallel computing, they have to be recombined into a single file. Then, the alignments are sorted by genomic location. After being sorted, the alignments are deduplicated. Finally, the alignments are filtered by various criteria, such as by the strength of the alignment and by the genomic location which the read aligned to. These steps must be carried out in that specific order. Sorting the reads first makes the deduplication process exponentially faster, so sorting takes place before deduplication. And to catch all duplicates, all reads generated by a biological sample must be examined compared to all other reads, so recombination, when applicable, must take place before sorting. The reason why deduplication should be carried out before filtration is a bit more complicated. It is because during sample prep for NGS, the data is extracted first before being amplified by PCR. Biological noise is present either before extraction or as a result of extraction, and that noise is amplified unevenly by PCR. Therefore, to calculate the true experiment efficacy, the data must first be deduplicated. Of course, doing these steps in a different order will almost never cause a significant difference in the resulting data metrics. Once the experimental noise and general biological noise has been filtered out, the data is now ready to undergo statistical analysis and biological interpretation. This is where the core pipeline ends, because the analyses which need to be performed are going to be experiment specific, and these can be very different from one another. Just to give a few examples, NGS can be used for gene expression analysis. In this scenario, the sequenced DNA is mRNA, and by normalizing the transcripts based on gene size and sample size, we can compare the gene expression profiles of different samples. In contrast, ChIP-seq experiments use NGS to sequence genomic DNA, which was extracted from a pull-down assay targeting a protein of interest. This allows us to deduce which parts of the genome the target protein is more likely to associate with. Very different. These are just two examples of the many experiments which can be done using NGS technology. But I digress. Now that we have gone over the whole pipeline in the first level of detail, let's dig a little deeper. We will begin with the FASTQ file format. As previously stated, FASTQ files store biological sequence data, with each entry consisting of exactly 4 lines: the read ID, the nucleotide sequence, any relevant line, and the quality scores of the nucleotide sequence. Let's talk about the read ID. Each read ID is, in theory, unique. No two reads in existence should have the same ID, since read IDs are normally generated by a combination of factors, such as the ID of the sequencing machine, the time and date of the sequencing run, flow cell coordinates, and so on. Read IDs are usually preceded by an @ character. The nucleotide sequence contained in the second line is fairly straightforward. It is a sequence of letters, with each letter corresponding to a nucleotide. A for adenosine, C for cytosine, G for guanine, and T for thymine. If the nucleotides at a given position is indeterminate, a placeholder will be used. For DNA sequences, this is typically an N. FASTQ files can also store amino acid sequences. I won't go over the one-letter codes for all 20 amino acid residues, but the placeholder letter for those sequences is typically an X. The third line is a bit of an oddity, in that, for the most part, it doesn't seem to have any uses. It usually begins with a plus character and can otherwise be empty. Sometimes the third line will begin with a plus character and then contain the same information as the first line of the entry, minus the @ symbol. Not much to see here. The fourth line is where it gets interesting. For quality scores, FASTQ files use a particular system. Instead of recording a number for each position in a human-readable format, FASTQ files use 1 character per position. Each character represents a different number, and those numbers range from 0 to over 40. It is done this way in order to reduce the file size and to make the file easier to compress. Because such files are mainly processed by computers, making the raw data easy for humans to read was a low priority when the FASTQ file format was being designed. So each character represents a number, but what do those numbers mean? The quality score numbers are known as Phred scores, and they indicate how likely it is that a given base call is incorrect. In most cases, Phred scores are calculated using the formula Q equals -10 times log P, where Q is the Phred score, P is the probability of the call being incorrect, and log is in base 10. In specific cases, such as the Solexa pipeline prior to version 1.3, the formula is Q equals -10 times log, open brackets, P over 1 minus P, close brackets, where Q is the Phred score, P is the probability of the call being incorrect, and log is in base 10. The key take-home in regards to quality scores is the higher, the better. A Phred score of 10 indicates a 1 in 10 probability of an incorrect base call, or an accuracy of 90%. A Phred score of 20 indicates a 1 in 100 probability of an incorrect base call, or an accuracy of 99%. A Phred score of 30 indicates an accuracy of 99.9%, and so on. If you are really interested, there is a Phred score conversion table in the appendices video. Phred score conversion tables are also very easy to Google. It is important to note that there are at least two Phred systems used in FASTQ files. Phred 33 begins at ASCII character 33, with the exclamation mark symbol corresponding to a score of 0, while Phred 64 begins at ASCII base 64, with the @ symbol corresponding to a score of 0. Therefore, it is important to first ascertain the Phred system being used before commencing analysis. The presence of certain characters would indicate that a file cannot possibly be using a Phred 64 system. They are shown on screen, but I won't read them all out loud. If none of these characters are present in the quality scores of a FASTQ file, then that would be a strong indicator that the file uses Phred 64. If a FASTQ file uses Phred 33 but doesn't contain any of these characters, it would mean that every single base call, without fail, had an accuracy of 99.37% or higher, which is unlikely. Okay, so let us now talk a bit more about the pre-processing which the FASTQ files undergo before alignment. I mentioned previously that there were three pre-processing steps: adapter and primer trimming, quality control trimming, and the combining and/or splitting up of files. To properly understand adapter and primer trimming, we need to understand the basics of the biochemistry underpinning the sequencing process. We will start by introducing inserts. Inserts are the nucleic polypeptides targeted for sequencing. They can be pulled down pieces of RNA, fragmented pieces of chromosomal DNA, or nucleic sequences obtained by some other means. During sample prep, adapters are ligated onto the ends of these inserts, forming what are known as fragments. Keep in mind that fragments, in the context of next-gen sequencing, means something completely different from fragments in the context of de novo genome assembly. In the specific case of Illumina sequencing, Illumina adapters can contain a binding sequence, a primer, and an index. The binding sequence allows the fragment to bind to the flow cell. Why is this necessary? Well, a single DNA fragment does not emit a strong enough light signal to be detected when sequenced. Therefore, all fragments need to be amplified by PCR. However, these PCR duplicates need to be grouped together to emit a cohesive light signal. This will not work if the DNA fragments float freely in the PCR mix. The solution to this problem is to have individual fragments bind to a solid surface via the adapter sequences, and the amplification duplicates bind nearby, also thanks to the adapter sequences. This allows small clusters of genetically identical fragments to form on said surface. The index allows fragments from different samples to be differentiated. When sequencing multiple samples in a single run, the inserts from different biological samples are ligated to adapters with different indexes. All samples are then pulled and sequenced on the same flow cell or flow cells. The index allows the fragments to be differentiated from each other. The practice of sequencing multiple samples on the same flow cell or flow cells is known as multiplexing. Multiplexing is done for two reasons. One reason is due to budget constraints, when using one flow cell per sample is deemed unnecessary and too costly for insufficient gain. The other reason is to hedge against the risk of individual flow cells behaving abnormally or failing. Finally, the primer is a binding site for polymerase proteins to bind to and makes the PCR process possible. Now that we know what a fragment is, we can talk about reads. When a fragment is sequenced, only the ends are sequenced, not the entire fragment. The sections of the fragment which get sequenced are called reads. In single-end sequencing, only one end of the fragments are sequenced, resulting in one read per fragment, while in paired-end sequencing, both ends of the fragment are sequenced, resulting in a pair of reads per fragment. These reads are designated R1 and R2 for the forward and reverse reads respectively. Because sequencing starts at the ends and progresses inwards, these raw reads will contain the adapter sequence. The adapters need to be removed, and the reads need to be sorted into groups based on their index. Most modern machines do this automatically, and if so, the FASTQ files they produce do not need to undergo this step. That is why adapter and primer trimming has been designated as a non-mandatory step in the core pipeline, because that step is not always necessary. One clarifying note about how the two reads are differentiated from each other. Each fragment has two ends. Different copies of a fragment will have different ends attached to the flow cell. The end which is not attached to the flow cell is the one that gets sequenced. What makes paired end sequencing possible is that the adapters at each end are different. When the forward read is sequenced, the exposed forward adapters are popped off, and the exposed DNA is then sequenced. The forward adapters bound to the flow cell are not exposed, and therefore untouched. Then, when the reverse read is being sequenced, the exposed reverse adapters are first popped off before the exposed DNA is sequenced. The quality control trimming and filtering step is much easier to understand. Basically, reads whose quality do not meet a certain standard will be trimmed down or removed entirely. Some trimming and filtering strategies include read length, average read quality, minimum base quality cutoffs for leading, trailing, and any base, and the sliding window strategy. Sliding window filtering involves calculating the average quality of all X-length subsequences of the read being scrutinized. Reads are either trimmed at the point where the average quality drops below the user-given threshold, or filtered out entirely. Sometimes, due to limited computational resources, data, even for a single sample, must be split into smaller files and aligned piecemeal before the results are recombined into a single alignment results file. Conversely, reads from the same sample but different flow cells may be combined into a single file to make the overall pipeline simpler, if computational resources allow it. These are logistical issues rather than biological ones. Similar to adapter and primer trimming, this process is not always necessary. Okay, now it's time to go through the SAM file format in much greater detail. As stated previously, SAM files consist of two parts, the comments and the alignment data. When it comes to the comments section, there's not much more to say here. As stated previously, these comments can be used to record notes and various metadata, just as they can be completely absent. Different alignment software and different SAM file processing tools each use their own system when recording metadata via these comments. We don't need to know what these systems are in order to understand the core pipeline, so there isn't much point going any further. When it comes to the alignment data table, however, there is a lot more to talk about. As previously stated, different rows are separated by new line characters, while different cells of the same row are separated by tabs. Each line represents an alignment of a read against the reference genome, but this is not necessarily a one-to-one relationship. One read in the FASTQ file can produce multiple rows in the results, i.e., multiple alignments. This is rarely done because the resulting SAM files could be enormous. Nevertheless, it is possible to specify that the alignment program do so. Let's talk about the table columns in a bit more detail. The first column, read ID, is fairly simple. It should be the same as the ID of the corresponding read in the input FASTQ file. The second column, the bitflags column, is probably the most convoluted and difficult to understand. To properly understand the bitflag system, we would need to understand basic computer architecture. In computer science, a bit is the smallest possible unit of information. It can be either 1 or 0. Typically, 1 represents true/yes, while 0 represents false/no. The information for multiple flags can be stored as consecutive bits. For example, 4 flags with the values of yes, no, no, yes would be 1, 0, 0, 1. If read as a binary number, this would be equal to the decimal number 9. In reverse, we could break the number 9 down into 8 plus 0 plus 0 plus 1 and reverse engineer the bit flags from that number. Theoretically, this allows us to store a lot of information with just a number which can save space, but in reality, this is stupid. It saves very little hard drive space, while at the same time, it is virtually the opposite of easy to read for humans. Using a single unique character for each active flag would only increase the memory footprint of a SAM file by a few percent, if that, and be much easier to read and interpret. But the established format is what it is, so we have to just deal with it. Now, before I explain what each of the flags are, I will need to explain what I mean by the word template. When I talk about SAM files in bioinformatics, a template refers to a biological sequence of relevance which consists of an individual read or the fragment implied by a read pair. In paired end sequencing, a template is a fragment which was sequenced and consists of 2 reads, located at the start and end of the template. The next read in template would thus mean the other read in the pair. In single-end sequencing, each read can be considered a template by itself, which makes the term somewhat redundant in that context. Now, let's talk about the bit flags and what they mean if set to true. The first flag indicates that this alignment was performed on a read which is part of a template which contains multiple reads. The second flag indicates that all reads in the template, which this read is a part of, map to the reference genome and in a sensible way. The third flag indicates that this read is unmapped. The fourth flag indicates that the next read in this template is unmapped. The fifth flag indicates that this alignment is against the reverse complement of the specified position in the reference genome. The sixth flag indicates that the next read in this template is mapped against the complement of the specified position in the reference genome. The seventh flag indicates that this is the first read in the template. For this pipeline, it would mean that this is the first read in the pair. The eighth flag indicates that this is the last read in the template. For this pipeline, it would mean that this is the second read in the pair. The ninth flag indicates that this alignment is not the best alignment for this read. It should only appear when users allow reads to yield more than one alignment result each during the alignment process. The 10th flag indicates that the read for this alignment fails platform or vendor quality checks. The 11th flag indicates that the read for this alignment was determined to be a PCR or optical duplicate, also known as sequencing noise. The 12th flag indicates that the read obtained via sequencing was split up into 2 or more subsequences, and this is the alignment result for a non-primary subsequence. And that should cover it. The 3rd and 4th columns denote the genomic coordinates of the alignment. The 3rd column contains the name of the chromosome which the read aligned to. The names in this column will be identical to the names used to generate the reference genome index. If the read did not align to anything, then this column will contain an asterisk. Meanwhile, the 4th column contains the chromosomal positional index of the alignment. The underlying concept itself is quite simple. Every nucleotide in a chromosome has a positional index. The very first nucleotide in a chromosome is base pair number 1. The second nucleotide is base pair number 2, and so on. The index number works like the coordinates of a one-dimensional map. When a read aligns to a part of a chromosome, it will align to more than one nucleotide, but there is no need to record the indexes of all the nucleotides. Only the leftmost positional index is recorded in the 4th column. Numerically, this will be the index with the lowest value, while biochemically, this will denote the nucleotide closest to the 5' end of the chromosomal DNA strand. It is very important to note here that data processed using different genome assemblies of the same organism cannot be combined. This is because the same DNA sequence can be given different positional indexes in different versions of that organism's genome. For a quick heuristic glance, data processed using one genome assembly can have its genomic coordinates converted to the approximate equivalent in a different genome assembly, using software such as Liftover. However, the reliability and accuracy of this translation is not guaranteed. For stringent statistical analysis, data should always be run through the same pipeline under the same conditions. The 5th column contains the mapping quality score, also known as the MapQ score. The MapQ score is calculated based on the probability of the alignment being incorrect. MapQ equals -10 times log p, where p is the probability that the alignment is incorrect, while log is in base 10. Long story short, the higher the MapQ score, the more reliable the alignment is. Keep in mind that the MapQ score is a comparative statistic. If multiple locations in the reference genome tie for the best alignment, then the MapQ score is 0, because we have absolutely no confidence in either of the alignments. Therefore, a high MapQ score does not just indicate good base pair matching, it also indicates a lack of competing alignment site candidates. The 6th column contains the compact, idiosyncratic, gapped alignment report, also known as the cigar string. The cigar string is a number of letters and numbers which report how a query sequence, in our case, the read, aligned against the reference sequence, in our case, the reference genome. A cigar string will contain one or more elements. Each element consists of a number and a letter. The letter, known as the operator, indicates the alignment result, while the number indicates the number of nucleotides. On this slide, I have described several cigar operators and what they mean. For those of you who are not familiar with database terminology, query sequence here refers to the sequenced read, while reference sequence refers to the chromosomal sequence. At least in the context of this current slide, that is what those terms mean. An M is used to denote nucleotides being paired up, whether that pairing is a match or a mismatch. A D denotes a deletion, or gap in the query sequence. An I denotes insertion, or a gap in the reference sequence. An equal sign denotes matching bases, while an X denotes mismatching bases. H stands for hard clipping, while S stands for soft clipping. They function like deletions, but at the ends of the query sequence. According to the official SAM file specifications, hard clipping should be used when a read is chopped up into subsequences which align to different parts of the genome and are recorded in separate entries. The chopped off nucleotides will not be present in column 10 of the current SAM entry, and will instead be present in a different SAM entry. This prevents the same nucleotide sequence from duplicating as a result of being present in multiple SAM entries. When the clipped nucleotides are not used in a different SAM entry, then soft clipping should be used. At least, this is how hard clipping and soft clipping should be used, according to the official SAM specifications, but some programs do things a little bit differently. Back to the commonly found cigar operators, P stands for padding and denotes a gap which is in both the query and the reference sequence. This is used for multi-query alignments. Finally, we have N, which indicates query nucleotides which align past the end of the reference sequence. It works like clipping, but for the reference sequence instead of the query sequence. This occurs when the reference sequence ends, but the aligned query sequence keeps going, such as when the read aligns past the telomeres. These are the main ones. There may be more operators out there, but they won't be covered here. On this slide, we have several examples. The main thing to note is how 6M can be used to denote 6 aligned nucleotides, whether they are all perfect matches, like in the first example, or whether they contain some mismatches, like in the second example. The 7th column and 8th columns function like the 3rd and 4th, in that they contain a set of genomic coordinates following the same set of rules. However, these coordinates are for the alignment results of the other read in the read pair. For single-end sequencing, where there are no read pairs, the 7th column will always contain an asterisk. Similarly, the 8th column will always contain a 0 for single-end sequencing data. The 9th column contains the size of the fragment that was sequenced. While this can be calculated from the other available information, it is convenient to have as a readily available data value, because we cannot obtain the size of a fragment by simply subtracting the index of one read from the other, as you can see from this figure. The 10th column contains the biological sequence, which is often, but not always, identical to the sequence in the corresponding entry in the FASTQ file. Similarly, the 11th column contains the quality scores of the sequence in column 10, and this is often, but not always, identical to the scores of the corresponding entry in the FASTQ file. The first 11 columns are standard across all SAM files, but more columns may be added which contain additional information. Different bioinformatics programs may put different information into these additional columns, and there is no standardized system for what information is included, nor how that information is formatted. I won't go through all of them, but I will give an overview of the one used by Bowtie, an alignment tool I am fairly familiar with. Under this system, each piece of information recorded consists of 3 parts: the tag, the type, and the value, separated by colons. The tag consists of 2 characters and specifies the bioinformatic nature of the information being stored. Examples of this include the alignment score of this alignment, how much weaker the next best alignment is, and so on. The type consists of a single letter and specifies the format or type of data. Examples of this include integer, string, char, and so on. Lastly, the value is the data value itself. For example, the tag AS signifies the alignment score, which is stored as a number. The type I indicates that the value will be an integer, so ASCII 10 records that the alignment has an alignment score of 10. Multiple such pieces of information are typically separated by white space characters. Keep in mind that this is just one of many systems for denoting additional information in a SAM file, and the one you come across in your project may be different. And that concludes the segment on SAM files. Finally, let us talk a bit more about post-alignment processing. More specifically, let us talk about the filtering step, since the other three steps are relatively simple and have been adequately covered with just the first level of detail. There are many criteria which can be used to filter alignment results. These include screening out alignments against artifact sequences, alignments against noise, ambiguous alignments, and weak alignments. Many of these results can be omitted from the raw alignment results by customizing the reference genome and by configuring various software options during the alignment process. However, rather than omitting these results from the raw alignment results, it is usually more informative to filter them out afterwards, because this allows users to calculate the percentages of the causes of disqualification. For example, in a gene enrichment pull-down experiment, including the mitochondrial chromosome in the alignment process allows users to differentiate between truly unalignable reads and reads which align to the mitochondria. Results which are disqualified from the main analysis pipeline may also still be useful in side analyses, or may be salvageable under certain circumstances. Since the alignment step is usually one of the bigger bottlenecks in the pipeline, it is more expedient to include such results in the raw alignment data and to remove them later. Also, while I use the word removal to describe the filtration process, what usually actually happens is that a copy of the file being filtered is created. In the copy, the disqualified alignments are not included. However, the original unfiltered results are still present in the original file, which has not been altered. Therefore, it is relatively easy to backtrack if you regret removing certain results, and you don't need to perform the time-consuming alignment process again. Now, while I gave an example in which mitochondrial DNA was considered noise, this is just one example of many. Different experiments will introduce different sources of noise, and different protocols will produce different experimental artifacts. There is no one-size-fits-all when it comes to filtering. However, removing experimental artifacts or noise is a relatively straightforward idea in and of itself. A trickier filtration issue to deal with is that of ambiguous alignments. An ambiguous alignment is when one read aligns to multiple locations in the reference genome. The 12th column of the SAM file may contain an XS tag, which indicates the score of the best alignment score other than that of the current entry. XS:(I:10) would indicate that there is an alternative alignment with an alignment score of 10. Keep in mind that this only applies to files generated by Bowtie 2. Other programs have been reported to use the XS tag differently, so take the information in this video with a grain of salt. In some experiments, reads which align to multiple locations may be tolerated if the score for the secondary alignment is far weaker than the score for the primary alignment. In other experiments, all reads with any degree of suspicion are removed. The appropriate course of action would depend on the experiment from which the data had originated. There are many other factors to account for when deciding on the filtering steps for the read alignments, but these are some of the more general ones. And that concludes everything I have to say about the core pipeline. But how do we get to the starting point of the pipeline? The pipeline I just described begins with FASTQ files, but how do we get them? Generally speaking, there are three ways. Firstly, they can be obtained directly from a sequencing machine after a sequencing run. Secondly, They can be extracted from an SRA file after downloading said SRA file. And lastly, FASTQ files may be downloaded directly. Out of these three methods, I especially want to talk about the first one, since sequencing machines may sometimes produce a corrupted FASTQ file, but the situation may still be salvageable. And to understand how, we need to learn a bit more about the sequencing process. When sequencing a sample, machines first generate a data file containing the visual output from the sequencing process. This visual output is then interpreted into sequencing data. Modern sequencing machines carry out this process automatically, so users typically don't have to worry about this step. However, on rare occasions, the interpreting process runs into an error and produces corrupt FASTQ files. If the problem lies in the interpretation step and not in the visual output data file itself, then the experimental data can still be salvaged by running the interpretation again on the visual output file. I also want to quickly address the question some of you may have, namely, why we download an SRA file and then extract FASTQ files from it instead of downloading the FASTQ files directly. The simple answer is that this is not always an option. Some databases, such as NCBI, store their data in the sequence read archive file format. In order to use data from these databases, we have to go through the process of first downloading SRA files and then extracting FASTQ files from them. Now let's talk about how to set up the pipeline. That is to say, how do we get all the files we need to run the core pipeline, aside from the data to be analyzed? Well, we first download the relevant FASTA files from an appropriate online database, such as UCSC. These FASTA files make up the genome of our organism of interest, with each chromosome's sequence stored in a separate FASTA file. We may also need to download the PhyX genome. The files are modified as necessary and then combined. This gives us a single FASTA file which contains the genome of our organism, as well as any other relevant sequences like the PhyX genome. Using this file, we construct an index. We now have an index for our organism's genome which we can align our data against. Let us go into a bit more detail. There are numerous online databases from which you can download genome assemblies. I would recommend the UCSC genome website. Now, this may not be the case for all online databases, but with the UCSC one, there are two ways to download data: via an internet browser or via command line. If you want to do it via an internet browser for the UCSC database, you start by going to the following URL and then scrolling down or using the search function to locate your organism of interest, as well as the assembly version you want to use. Once you've found the assembly you want, click the Sequence Data by Chromosome link. That should take you to a page with a similar URL to this URL on screen, and have an appearance similar to this screenshot. Scroll down until you see the download links. Click the links to download the FASTA files of the chromosomes you need. Make sure you remember to include the mitochondrial and sex chromosomes if they are relevant. As for downloading files via command line, instructions can be found at the link shown on screen. An introductory guide to command line has been linked in the video description. Now that we have gone through the downloading process, let us go over what exactly it is that was downloaded. The FASTA file format is a format created for storing sequence information. The entries stored can be as short as a few nucleotides, in the case of primers and restriction sites, or they can be as long as hundreds of millions of nucleotides, in the case of chromosomes. Each entry consists of two parts: the ID line and the sequence, written across an unspecified number of lines. Therefore, unlike FASTQ files, which strictly follow the rule of 4 lines per entry, in FASTA files, there is no set number of lines per entry. Let's talk a bit more about the ID line in FASTA files. ID lines are lines in the file which begin with a greater than symbol, followed immediately by the name or ID of the entry. Additional information and annotations may also be stored on this line, separated from the ID by a whitespace character. All lines between that ID line and the next ID line are regarded as containing sequence information. With genomic sequences, this will generally be a sequence of A's, C's, G's, and T's, with unknown nucleotides being represented by N's. The sequence itself is typically in uppercase. Nucleotides can be soft masked by being recorded in lowercase, or they can be hard masked by being replaced with N's. There are a multitude of reasons for soft masking or hard masking data, but we won't go into that here. Let us go back to the chromosomal FASTA files we downloaded. They contain one chromosome per FASTA file, so they can be referred to as chromosomal FASTA files. Most chromosomal IDs are given using Arabic numerals, although some organisms, like C. elegans, use Roman numerals. Now that we know what we are working with, we can talk about the modification and combination step. Sometimes, the data in the FASTA files need to be modified before it is suitable for your analysis. This might be something as surface level as changing chromosome names, or it could involve changes to the sequences themselves. Whether or not the information in the FASTA files is modified, it is strongly recommended that all relevant files are concatenated into one file containing the sequences for all the chromosomes, also referred to as a genome FASTA file. Depending on the experiment, you may or may not wish to include mitochondrial chromosomes, X and Y chromosomes, and or the genomic sequences of potential experimental artifacts as pseudochromosomes. Making modifications and edits to the chromosomal data is usually unnecessary for well-established organisms such as mice and humans. For cases where modifications and edits are warranted, there are simply too many to be comprehensively covered. When it comes to renaming chromosomes, this can usually be done by opening the file in a minimalist text editor like Notepad or TextEdit, and then directly editing the file. Alternatively, command line can be used. It is also possible to use these text editors to combine files into one for the combination step, but it is recommended to either use a command in command line or a simple self-made script. For the first option, please refer to the command line guide, while the second option requires rudimentary programming ability. After the modification and combination step, we now have a single FASTA file which contains all the chromosomes we need for our reference genome index. Indexing is done to exponentially speed up the alignment process. This is because it is much quicker to look something up in an index than it is to trawl through the data. Therefore, the alignment step in the core pipeline actually aligns the reads against the index and not the genome FASTA file itself. How an index often works is that all subsequences of length k, also known as k-mers, as well as the positions of these subsequences, are recorded. When a read is aligned against an indexed reference, a k-mer is derived from the first k bases in the sequence. Afterwards, rather than comparing the query sequence against the entire reference, only the k-mer locations are examined in closer detail. The program may still have trouble aligning reads when there are thousands of instances of the same k-mer in the reference genome. If this occurs, it will be noted in the alignment results. Once the indexing is complete, we would have a series of index files with names based on the genome FASTA file. Collectively, these files comprise the index against which the alignment happens. And that concludes the core pipeline and all supporting pipelines. But wait, there's more. For those of you who don't have a background in computer science, I have included a brief explanation on binary numbers, how they relate to bits, bytes, and bitflags, as well as an overview of ASCII. Once you understand ASCII, the Phred score values will make a lot more sense. So, binary numbers. In the decimal number system, which we learned growing up, each position of a number can have one of 10 values: 0, 1, 2, 3, 4, 5, 6, 7, 8, or 9. When a position is at its highest possible value, incrementing that position causes that position to reset back to the lowest possible value, and the next position increases by 1. For example, 19 is followed by 20. Increasing the number 19 by 1 causes the 9 in the 1's position to go back down to 0, while the next position, the 10's position, increases from 1 to 2. Now, because there are only 2 possible values at each position for binary numbers, instead of counting like this, they count like this. And for anyone who is just listening to the audio but not watching the video, I just showed 2 series of numbers on the screen. Now that we know how binary numbers work, let us talk about bits, bytes, and bitflags. In the early days of proto-computers, a signal on a chip or wire's electrical circuit could either be high voltage or low voltage. Memory components, either temporary or permanent, would store this information, and thus only have 2 possible states. Such a system naturally lends itself to a binary number system, with the 2 signals being designated as either 1 or 0. The smallest unit of information in computing came to be called a bit, and it is a unit of information with only 2 possible values, 1 or 0. A byte consists of 8 bits and has 256 possible values. This limit underpins many things in modern computing. It is why some programs cannot parse files with file paths exceeding 255 characters in length. It is why RGB values go from 0 to 255. And it is why the Pac-Man arcade game glitches out after level 255. But going back to the topic of bytes. A byte is usually the smallest unit of information that code will deal with. However, if the information being stored is either yes or no and nothing else, then we only need 1 bit to store that information, not 8. The other 7 bits are redundant. Now, in the early days of computing, data capacity was much lower and more expensive, so there was a strong incentive to utilize every bit in a byte to store information if possible. This still holds true today in very specific scenarios. Ultimately, this led to the creation of bitflags, which are bytes where every bit counts as a separate yes-no piece of information. So how does this all tie back to binary numbers? Well, all bytes can be read as a binary number, and this includes bytes used for bit flagging. For example, a bit flag byte with yes for the first, fourth, and eighth flags, and no for all others would be 1, 0, 0, 0, 1, 0, 0, 1. If interpreted as a binary number and then translated back into decimal, this would be 137, which can be broken down into 128 plus 0 plus 0 plus 0 plus 8 plus 0 plus 0 plus 1. 1 byte contains 8 bits, and can therefore contain the data for 8 bit flags. 2 consecutive bytes will, between them, contain 16 bits, with a highest value of 65,536. You may also recognize this number from various places. Now that we understand how bits, bytes, and binary works, we can finally talk about ASCII. In pure data form, all information, such as letters and symbols, are stored as ones and zeros, which can be read as a binary number and converted into a decimal number. Here is an ASCII table, which you can find with a simple Google search. Under the ASCII system, lowercase a is stored as 0, 1, 1, 0, 0, 0, 0, 1, which corresponds to the number 97 as a decimal number. Uppercase Z is stored as 0, 1, 0, 1, 1, 0, 1, 0, which corresponds to the number 90 as a decimal number. The character for 0 is stored as 0, 0, 1, 1, 0, 0, 0, 0, and not 8 zeros in a row. The binary form of the ASCII character for 0 corresponds to the number 48 as a decimal number. And for our last example, the symbol for hash is stored as 0, 0, 1, 0, 0, 0, 1, 1, which corresponds to the number 33 as a decimal number. And now, at last, we can talk about Phred scores in FASTQ files. As stated earlier, FASTQ files use a single ASCII character for each nucleotide to designate the corresponding quality score. Similar to an ASCII table, there are two Phred tables which tell us the corresponding quality score for each ASCII character. Be mindful of the fact that a quality score of 0 does not correspond to the ASCII character for 0. Under the Phred 33 system, a Q score of 0 is represented by the ASCII exclamation mark, which has a decimal value of 33. Under the Phred 64 system, a Q score of 10 is represented by the ASCII uppercase J, which has a decimal value of 74. And so on. Like ASCII tables, Phred tables can be found using a simple Google search. And that concludes the presentation, appendices and all. Slides and supporting documents can be downloaded from the links in the description below. My name is Angelo Chan, and I hope you enjoyed this video series.